Hello, and welcome to the BOFIN Buzz, the podcast for anyone fascinated by farming's future. Well, hello, everyone, and welcome to the BOFIN Buzz. And I'm just here to say that we're going to be talking a lot about the Truth Project. The website address is truthproject.uk. That's truthproject.uk, not truthproject.co.uk, which is how I refer to it in the episode. But anyway, on with the episode. Enjoy. Well, hello again, and welcome to the BOFIN Buzz. And I'm delighted to be joined today by Dr. Maria Hernandez-Soriano. Hello, Maria. How are you?
Hello, Tom. I'm good, thank you.
Great, fantastic. Well, look, Maria, I know Maria because we're doing a project together that you'll hear about in a bit. But what I'm really looking forward to in this conversation today, Maria, is talking about science, and in particular, some of the science that you're getting up to at the John Innes Centre. So we'll look into that in a little bit more detail. But first of all, can I just ask you if there were three key things that, in your view, are important to address in UK agriculture? What do you think there'd be?
Well, it's definitely a key thing to improve the health and the productivity of our soils. And of course, that is immediately linked to the health of our crops. And
Absolutely.
secondly, we need to change a bit our focus in how we see agricultural systems, what do we expect from them, and how we can advance them towards a more resilient, because we are having a lot of problems at the moment. We're having climate change and
Mm.
loss of productivity. So we want to rechange a bit, refocus a bit our attention on how we want to address the problems of agricultural systems. And of course, we want to, as a scientist, what a part of my work is to provide tools, to provide the capability for producers, for farmers, to actually be capable of assessing how productive their soils and their crops are.
Though that's fantastic. I have to say, they are three topics that I'm very interested in as well. And I know a lot of our listeners are as well, Maria. And of course, it's tying together those things with the science as well, which I think is really exciting, which I hope we can explore a bit more in this. But first of all, just want to hear a bit more about you. I mean, first of all, Maria, would you just like to introduce, who you are, what your current role is at the John Innes Centre, please?
Well, my career has gone kind of uphill in achievements of what I wanted to do since I was little. So I got an interest in agriculture when I was little because I spent the summers with my granddad. We have a little orchard, and I was just fascinated about how the vegetables were growing. That was my
Where was that?
back in the south in Spain.
Ah,
I come from Granada, it's a city in the south of Spain, and in the summers I would just spend my summers in the little orchard, just growing peppers and potatoes and whatever we could figure. And then, since I was little, I have a curiosity about understanding how things work in nature. So I studied biochemistry, which was 5 years back in the south of Spain in the University of Granada, just learning things in the lab, just all this kind of a very molecular, microscopic level. And then the opportunity came to do a PhD in soil science, looking at the pesticides and irrigation and so on. And I didn't have a clue about soil when I started, but I think the first 2 months in my PhD, I absolutely was fascinated. Different types of soil, what much, how much goes on in soil. Chemistry, biology, everything I had studied suddenly makes sense because I could use it to understand what was going on in soils and particularly in agricultural systems. So from the very beginning, I was incredibly engaged in talking with farmers and seeing what they were doing and how was affecting the soil. So I moved to Belgium because in Belgium to make my PhD because in Belgium they have a strong interest in agriculture and environment. So it was kind of the best place to do a PhD for me.
okay.
And then I realized that it was not only the soil, that we need to understand the plant and soil interaction, that's
Right.
where the action is happening. So I completed a PhD. I learned so much about soil and about how to understand agricultural systems. I moved to Australia to understand other ecosystems. I went to the States to learn more techniques. which was really fascinating because
Mm.
the way agriculture is viewed in the States is really interesting. It's an absolute passion at all levels. And then I ended up then I moved to England for a number of reasons.
Yeah. So
And
when was that then, Maria?
well, I moved here in 2017, and after a bit of reshaping my career, because I have been focused in a very lab scale, only in Australia I started to work more in the field. And then the opportunity to work at John Innes came in 2019, and it's been 5 years. And well, I have to say, my very vision of how I wanted to do science has been fulfilled here in the sense that I can do experiments in the field, looking at plant soil interaction, but also John Innes has a strong drive into interacting with farmers because everything we do in the institute is to improve
Yeah.
agriculture. Is our main drive. So,
So
and
you
here I am. Now I'm working in a big project into
yeah.
screening wheat and basically looking for traits that can improve both the productivity of soil and the crop itself.
Yes, so this is the Wish Roots project. Is that
Yes, yeah.
right? Yes. Tell us a bit more about the Wish Roots project. So this is taking place. Well, your role within this is at John Innes Centre.
Mhm.
That's right, I think. What's your actual your role here at John Innes Centre?
Well, I'm a postdoctoral. That means that I'm running this project.
Right.
And we are coordinating it. So it means that we have 7 other partners in different countries. So from
Right.
Europe, China, South Africa, Scotland as well. And the main, so WISH roots stands for Wheat Improving Soil Health through Root Traits. So that means we have selected a number of cultivars of wheat, modern ones that are currently in the market, but we also have something precious at Unines. Is kind of one of our treasures. Is this collection of 1,000 wheat land races that
Right.
are historic land races. They are not grown anymore commercially, but
Sorry,
they
this
have.
is the Watkins lines. Is that right?
This is the Watkins lines.
Yes. Now, our listeners will hopefully be familiar with the Watkins lines because we've got one or two projects already going on
Mhm.
within BOFIN looking at the Watkins line. And in particular, many of our listeners will know about the Watkins 788, which we're looking at in our slug project. Listen out for some results that we'll be publishing soon about those. We've got some feeding trials going on underway at John Innes Centre as we speak in the insectary, looking at those. But these are some other Watkins lines, I
Yeah.
think you're talking about. So tell me a bit more about those, Maria.
So during the last, previous to this project, my work at Ioninus has been focused on nitrification, also nitrogen, improving the efficiency for
Yeah,
plants to uptake the nitrogen. But we know how important nitrogen is for the plants, for growing, and for the quality of the seeds. So the lines I'm working with has been selected because our work has shown they have a capability to improve the control that the plant has on the nitrogen in the soil once we put the
right.
fertilizer.
Yeah,
There are a lot of microbes there that they do their own job, but the plants, certain of these Watkins cultivars, they are very good at controlling that nitrogen transformation, and that translates into an increase of nitrogen in the seed, which obviously improves the quality, but also is more cost efficient because the plant makes more of the fertilizer that we apply.
so hang on, just recap there because my knowledge of how it works in the soil is patchy at best. I mean, we know that we apply nitrogen to the soil, and there's a little bit of magic that goes on, and that makes it available to the plant, and that's to do with the microbes in the soil. Would you just like to talk us through that, Maria?
Happy to. So basically, there is always a pool of nitrogen in the soil naturally, but most of it becomes in a form. So the nitrogen can be in very different forms. So the form that the nitrogen end up in the soil is not available for the plant. It's organic. So
Right.
it's just there, but it's not available for the plant. There are there are thousands of microbes in the soil. Some of them are capable of transforming it, what it is natural in the soil, and make it available for the plant. The fertilizers that we put, they are already inorganic. They are ammonium and they
Right.
are nitrate. And those forms that we give to the plant is easy to take. It can be immediately taken. So the ammonium is very good for the plant because it doesn't take much energy to take, and it stays longer in the soil because it interacts with the minerals in the soil, so it kind of stays there a bit longer. So the plant has time to take it.
Okay.
However, we have a different guild of microbes in the soil that they transform the ammonium into nitrate. And
Right.
once it's transformed to nitrate, it's a bit more energy difficult for the plant to take. It's still good, it can still take it, but the problem is that it's incredibly soluble. So it goes very quickly into the water, and
Right.
later on it gets further transformed and it also becomes nitrous oxide. But as we probably know,
Yeah.
it goes into the air, it's terrible greenhouse gas, and at the end of the day, it's a loss for whoever has paid for the fertilizer because either into the water or into the air, it's gone.
Yeah.
So it's
Yeah.
bad
And
for the
of course,
environment, bad for the
very
pocket.
that's very topical at the moment with climate change and nitrous oxide being one of the worst gases, I think, for global warming. And this, so this is down to our use, mainly down to our use of nitrate fertilizer, ammonium nitrate fertilizer, I guess. So yeah, just to sort of clarify, this is so the fertilizer goes into the soil. And it's the microbes within the soil that make it either into nitrous oxide or into nitrates that leach out. Um, but what do the plants do then? And what's going on in the soil that stops that happening?
Well, our all plants are very clever on that because all these land races from the Watkins that I have narrowed down, we have a
Yeah.
bunch of like 10 or so, they react. They react to the application of the fertilizer, and then they also they immediately control these microbes that do the transformation from ammonium into nitrate. And by control, I mean they just release something into the soil that at the moment we don't know yet, not
Right.
for wheat. We know for other crops, but we don't know for wheat. But we have made the link that the moment you apply the fertilizer, instead of these bacteria, these microbes getting kind of multiplying and starting really, really quickly to transform the nitrate, these land races have the capability of stopping that transformation. They control this bacteria.
Right. So this is. Am I right in saying that this is biological nitrification inhibition? Is that right?
Exactly.
So, and so these are the plant. This is incredible, Maria. So these plants have the ability to put something into the soil. We know not what it is yet, but put something into the soil that slows down the bacteria and this process within the soil that's causing pollution. I mean, this is huge, isn't it? We're talking about an ability within wheat, but you say this is what you found in the Watkins wheats.
Mhm.
Okay, so I'm gathering from that it's not in the wheats that we're using at the moment, not in the commercial wheats. Is that right?
That's correct. What we see in the modern varieties is that when we apply the ammonium fertilizer, there is an incredible rise in this bacteria. Their abundance just spikes up, and the plant doesn't seem to, the modern cultivar seems to have lost that capacity to control
Right.
it.
Gosh, I mean, that is huge. So if we can get that trait from those Watkins lines into our commercial wheats, then we can potentially, solve the whole pollution problem, really, can't we?
It will definitely make a huge improvement because the plant will take more, will have more time, will gain more time to get the fertilizer.
Yeah.
And obviously,
Fantastic.
we will decrease the need to apply so much fertilizer, and we will definitely decrease the amount that is lost to the environment.
Yeah. So in the same route, we're going to be improving nitrate use efficiency, aren't we?
Exactly.
Ah, fantastic! I mean, this is a win on so many levels. It's so exciting, and I can't believe that we've got this sort of sitting in our genetics. The question is, can we get it from the genetics of these Watkins lines? I mean, tell me about these Watkins lines. Are they, would I want them in my fields, for example?
Not particularly. They have a love for lodging.
Right? What they lodge a lot, do they?
And they have these owns. They
Uh
have
huh.
fantastic owns that they were once in a time they were useful for the plant when they were wild, but at the minute it makes harvesting and processing the very difficult. So it's not a cultivar you want to grow
Right.
per se, but we can identify where in the genetics of the plant this is and add it as many other things we have added into the modern cultivars.
So hang on, have you managed to this ability of the plants to do its own biological nitrification inhibition? Have you narrowed down where that is in the genome? Then,
We are in the process. We have located the genome of the genome of the wheat is complex because
yeah,
with all the crosses and breeding it has become very complex. But a couple of years ago we confirmed a few of the locations,
right.
and now we are in the process of exploring either how to bring it just by classic crossing, or we could even just we have no better technologies to accelerate the process and chop
I
this big
see.
and put it into the new ones.
Okay, this is really exciting. So now, am I right in thinking that these are the quantitative trait loci or something? Is that right?
Quantitative trade loci, very good.
Yeah, or QTLs. So that's places within the genome where the magic is happening. The genetic code is in there somewhere, but you haven't narrowed it down. So it's a bit like knowing I don't know which sea or which lake in which to fish for the right fish, but you haven't actually found the fish yet.
No, the fish is at the moment the way we do it, it gives us very big chunks. So we have to kind of go into the detail
Okay.
to see if we can reconstruct what is the pathway, the route that the plant uses to produce these compounds in response to the fertilizer and the increase in these microorganisms.
So why can't we? I mean, can we just? I guess there's GM techniques, for example, where you
Yeah.
could just take that whole chunk of genome and stick it into another wheat. But that's GM because we don't want that because we're not going to be able to grow GM. But could you not cross it? Cross that wheat with with a modern wheat, and then have a look at the genome, and presumably, if you know where the quantitative, the QTL is, if that's come over into the new variety, hey presto, we're away. Why don't you just do that?
There is that's an option, of course. The problem is that it's even if we narrow it down. So the way we do it, we take the two parents, the one that does the trick and the one that doesn't, and we
Yeah,
make a number of crosses. So we make 80 crosses and we grow them, and then we make a link in between which of the crosses do more the trick than other, and that's how we
yeah,
find out these chunks. Then we can select these particular crosses that seem to be the best candidates, and then the breeding companies can start doing other crosses to see when the trait is shown. The problem is that it takes a lot of time. It can take
yeah,
several years to do. Using GMOs, of course, it will accelerate. Using some of the modern techniques that you can literally just chop something and put it into the new one. Let's remember, we are not talking about introducing anything new. We are
no,
just restoring something that was at some point was already there. It's absolutely natural. So it's not
yes.
introducing anything strange in the genome. It's just bringing it
So,
back, basically.
so there's probably something. I mean, in modern wheats, they've presumably got the same genetic architecture. It's just a question of once you know precisely within the genome where that is, uh, can you? Because we've we know about gene editing, and we've talked about gene editing before on this podcast. I think, but as I understand it, this is the sort of thing that we could do with gene editing. Once we know precisely where it is in the genome,
Yeah.
we can make a small edit. Isn't that right?
Yeah,
To make it
in
to
theory, it's that simple.
yeah. Um, but we need to know precisely where that is first, and that work hasn't
Yes.
been completed.
No, and also it turns that, well, it's not always just one chunk. You will
Yeah.
find that the production of these kind of compounds is not the primary work of the plant. This is something more special, it's more specific, more specialized.
Mm.
So it normally takes few chunks within the genome. So you have 6 chromosomes, 3 times repeated, seven, sorry.
Right.
So it makes things complex. because sometimes you don't have a copy in all the three repetitions. And
Mm.
then it turns out that to produce a compound that specific, you find that it's not only just one region. You will have something in chromosome 2, something in chromosome 3, and something in chromosome 7. And
Right.
some three different bits that work together to put this compound in the soil. So that makes it just slightly more complicated.
Yeah, I know, and I think if I remember rightly, so we've got just one copy of genetic code, I think, haven't we? Whereas wheat is a hexaploid, and so it's got three copies. And I think someone told me the other day, 17 billion letters that you've got to sift through to find the right ones that turn on and off these genetic bits. I mean, is this doable, Maria? Do you think we'll find exactly? And do you think we can do it through gene editing? Because we now know that there are we can now grow gene edited crops in the UK as long as it can be as long as it's an edit that could happen naturally. I believe is
Exactly.
that right? Yeah. So I mean, when they say it could happen naturally, Maria, what does that mean?
It could totally happen naturally in the wild. I mean, it's just something. In this case, for instance, these land races, these Watkins lines,
Hmm.
they are originally from Iran, Iraq, all this area, and this capability of the plant was developed, naturally evolved in response to the lack of nutrients. So the
Right.
plant evolved naturally to do that. So again, it's not something, and that's where the wheat was first cultivated. So it's the very origin. So it's something
Yeah.
incredibly natural. It's just that over breeding over the last 100 years or so has focused on yield, easy harvesting, so on. And because we were doing it just like by crossing for what was looking better or producing more, we have just accidentally lost it.
Yeah. So
It's
what
just a
could be
matter of
more
bringing
natural?
back.
Uh, yeah, I suppose what could be more natural than switching back on a gene or switching off a gene or whatever it is that you do, that it gives it this natural ability that a wheat had, but has been lost in our modern wheats. What could be more natural than restoring it, if you like, restoring the capability? But I want to turn now because uh, I know that this is something that we're looking at in a new project, and we're really excited about this project, aren't we, Maria? So, just to introduce it to everyone, this is Truth, which is Thriving Roots Underpinning Total Soil Health, and this is a new project. It's a project that we've got. I think it's five industry partners. So BOFIN is leading it. We've also got the University of Nottingham. We've got John Innes Centre, of course. And we've also got PES Technologies. And the really exciting thing about PES is that they've got this new soil nose. So it's a sensor that can smell the soil and it can sense the volatiles that come off the soil and tell us an awful lot about it. Now, where they've already calibrated the PES sensor for various qualities of the soil, but what we're looking at within this project is calibrating this sensor for the soil microbes, the sorts of things that we've just been talking about. And we know that it can sense the difference in soil microbes. What we're going to be doing in the project is doing a lot of testing with farmers, and we'll talk in a bit about how farmers can be involved, so that we can calibrate this sensor, and it can be an in-field quick tool that people can use to actually gain an awful lot of understanding about our soils. So, could you just that's a brief introduction to the project, but Maria, we're looking in some detail at some of the wheats that you're bringing through in the Wish Roots project under the Truth project, aren't we? Would you like to just talk a little bit about that?
Yes, so we have selected, I mean, we have now more information about these land races, these cultivars, as we have been progressing with the wish roads. And we have selected the best candidates that we know they have the most stronger capability to control this transformation of the nitrogen. And through the truth, we are going to be testing them. I mean, at the moment, they have only been tested in our field trials at Jonines, but the truth is kind of the best heritage that the that the waste roads can have, because now we can upscale it to actually bring it to farms, different soils, different land managements, and see if they actually keep having this capability in all these different settings. And we are going to be looking at the microbes as we have been doing. So in collaboration with the root rangers to be,
Yes.
we will be collecting them through all these different field trials, and then I will be looking at the microbes and how the different cultivars are affecting them. And we will be cooperating with PES. So this information that we are gaining from my side, looking at the microbes, then PES is going to use it to calibrate, to kind of tune their instrument, the
Yep.
soil nose,
Yes.
to advance a bit the capability of these tools to be just deployed in the field and quickly give us an idea of about how the plant is controlling the
So,
nitrogen.
so what you're doing, so that I think there are two aspects to what you're doing within the project. First of all, you're going to be taking the soil samples that we receive. Oh, one thing I should point out is that one of the main things that we're doing within this project, and I should have said this at the start, is we're going to be appointing root rangers. And this is a feature of all of our projects at BOFIN, that these are farmers who are going to be paid to do on-farm trials as part of this project. So we're looking to set up root rangers who are going to be our in-field experts, if you like, or they're going to be our in-field experts by the time we finish this trial, because there's going to be a lot of training involved. The idea is, so the root rangers are going to be looking at an area in their wheat crop and sampling that, taking samples from that. And we're talking about specific samples, looking at specific samples that you're going to be analyzing in the lab, looking at the microbial content. And it's not just the soil itself you're interested in, I think, is it? Tell me a bit about that.
Exactly. It will be if specifically the soil that is in very close interaction with the roots.
Yeah,
We
in
call
the
that
rhizosphere, isn't that right?
we call
Yeah,
it the rhizosphere, and particularly we call it the rhizosheath, the soil that really grows adhere to the plant, because this is where all the action happens. This is where
right.
the uptake of nutrients is happening for the plant,
Yeah,
let's say the nitrogen, but it's also where the plant has this point of deploying whatever compounds they are putting through the roots to control the microbes. So
right.
we want to look at that specific bit of soil that is stick to the root.
Great. Yeah, and what we're looking to set up, and what we'd really like those people who are interested in this to do is I think we've set up a new website address, which is truthproject.co.uk. So that's truthproject.co.uk. If you go to that website, you can find out how to get involved. And significantly, if you'd like to be a root ranger, one of our paid farmers who will be doing the trials. And what we're looking to get here is a range of soil types, a range of locations, a range of farming systems as well, conventional, organic, as well as regenerative farmers, so that we can get a really good mix of natural arable systems, if you like. And we'll be asking those farmers to send in samples to Maria, so that you can analyze them, get a really good idea of its fingerprint, if you like, its microbial fingerprint. We'll then compare that with the samples. No, we'll use the same samples through the PES sensor so that we can calibrate the sensor through the course of the project. That's the idea. So keep a lookout for that. Keep a lookout for the emails that we send out about the Truth Project as well, and how you can be involved and how you can get a really good understanding of what's going on in your soil, and the microbial signature of it. And we'll be publishing more details about that as well. So now, within this, one of the really exciting things about this, though, Maria, is that now we were talking about your Watkins lines earlier on, and just tell me how much of that material have you got at the moment? Have you got enough to put into farmers' fields for them, for example?
Not this year, we need to multiply.
Yep.
So we have a few kilos because obviously, our trials are normally quite a small scale. But we are talking here of a quite exciting, big scale that is really, really representative. That's the
Mm.
beauty of the truth, I would say. And so this year we are already getting ready to multiply it. So we are hoping that by the third year we will have. So this year will be multiplication, next year will be a bit more of multiplication, and then the last year of the truth will see enough quantity for all the farm trials to be running.
No, well, that's really exciting. So, in the third year of the project, our root rangers will not only be looking at the microbial activity of their soils within their current wheat crop, but they'll be able to have a comparative strip next door to their current wheat crop of one of these exciting Watkins lines, and so they can see how the two perform and whether there's any differences. And that's going to be a game changer, Maria. I think it really is. Now it's not just the Watkins lines. I think we're looking at. We're looking at routing architecture as well. I think, is that right?
Yes, that's really exciting for me as well, because obviously the roots have quite a number of roles in the soil. I mean, they shape the microbes, and we are talking about nitrogen because it's one of our focus, and obviously it's very important.
Hmm.
But there are so many different functions of the microbes run into the soil that the plant can control. Just putting these different compounds in the soil, they are so specialized, and they are so dynamic and kind of flexible in the terms of how they can react. So that's one part of it. But obviously, as the root grow, we can just see it in our imagination. As the root grows, it shapes the soil. It holds it together. It makes pores. It makes a really good house for all the living things in the soil. But it also kind of allows good aeration, like all the air coming in, water holding capacity, so the water just doesn't go. It actually stays there because obviously every living thing in the soil needs water. The plant needs water, so that's really important. So we are going to be looking with the truth. That's something that we are also looking at in the waste roots, is how the architecture of the root, all these hairs, all these kind of angles in which it grows, the length, all the different characteristics affect the soil structure. And then our colleagues in Nottingham have quite advanced approach to do this, so they can look at very intact cores, as the plant grows, how the soil is being shaped, and all these pores and all this nice structure, which, by the way, it also affects how carbon is stored and all the organic carbon and carbon storage we are always discussing. So it's all interlinked. And that's
Yeah.
one of the big components also of the truth, is looking at comparing a bit the root structure with uh, the structure
Yeah.
of the soil.
Yeah, so and that's why Nottingham's involved, because they sort of lead the field in this respect, I think, and they do these CT scans of the cores, don't they? And I've seen some of the videos they've done of the computer graphics. They can make these fascinating. You can actually see what's going on with your roots, because that's the problem, that we know so much about what's going on above the ground now with our crop canopy and biomass and understanding of GAI and so on. We have very little understanding of what goes on below the soil. And I saw a paper the other day, for example, that said that modern wheats, they've looked at the rooting ability of modern wheats, and actually over the past 40 or so years, we have reduced the rooting ability, because we've bred so much for what we can see above the ground. We've forgotten about what's going on below the ground. But and this is what I wanted to ask you about, Maria, because I think, isn't it right that you've been looking at some of the architecture of these lost wheats again, the rooting architecture, and you found one or two interesting varieties. Tell us more about that.
Well, yes, in collaboration. So within the Wish Roots, we have a collaboration with some partners in Italy that have a few decades of doing research in root architecture,
Mm.
and that was bringing them into the project has been very useful, because obviously they know everything about the architecture, but for the first time with the Wish Roots, we have linked it to the aggregation, and aggregation is a soil that has good aggregation is not dust. It's soil that is spongious, and it's kind of has a nice structure and holds the water and the nutrients, and has good
Yeah.
porosity, which is essential. So last year was our first trial looking at the link. So we were looking both at how aggregated is the soil, and we were linking it to the architecture of 10 different varieties of wheat. In this case was
Nice.
durum. Be-
Durum wheat.
yeah, our partners in Italy are kind of focused on durum, obviously, because it's for pasta.
Yep, of course.
So they selected 10 cultivars of durum that they know everything about the architecture. So So we have a list that tomorrow in our webinar we are going to present this list and
Right.
how some of these are more adapted for drought, some of them are more adapted for taking nutrients, and it's all based in this architecture. But it's the new things that we are about to release this year from the Wish Roots is how some of them actually improve, as we expected, this architecture translate into better aggregation in the soil. It
Right.
was done within the Wish Roots. It was done a very kind of classic, very conservative approach. The truth is taking us a bit further because we are going to use the technology that they have at Nottingham, which is more kind of the you don't have it's a non-destructive approach. You can just take the soil intact and look at the structure in itself, and that's something we really all look forward.
I see. So some of these Durham wheats, then, what you're saying is that they've got a really interesting root architecture. Now, I'm
Very
just wondering, do you think we can have a look at some of these Durham wheats within the Truth Project? What do you think?
absolutely.
Yeah. So perhaps that's something we can introduce again in the third year of the project. Some of our route rangers might want to look at the Watkins lines. Some of them might want to look at the Durham. Perhaps there'll be enough material that they could look at both within the same trial.
Yeah,
That's something to
that's
explore.
the ongoing
Now,
plan.
no, and one of the other aspects where we talked a little bit about so the our route rangers will not only be taking the soil samples that we talked about earlier for Maria and for the PES sensor, but they'll also be taking soil core samples. This there's a method of doing this that we've developed with the University of Nottingham, and we'll be giving farmers training in how they do this. So that's something that we're going to be rolling out over the next month or two. Do keep a lookout for how you can be involved in this, and that's going to involve sending soil cores back to Nottingham to get this really interesting sort of analysis of what your roots are doing. In this first year, obviously, it's going to be with just the varieties that farmers have got in their fields at the moment. But then next year we're going to be in the last year, as we've talked about, we're going to be growing some of these really interesting wheats that Maria's got up her sleeve. And then next year, what we're hoping to do, I think, is look for varieties that are widely available that have an interesting root characteristic. Am I right? That's what we're doing in the second year, isn't it? Now, how are we going to find those wheats, Maria?
Weeds that have an interesting architecture.
Yes, so wheats that have got an interesting root architecture or something interesting about them that we can introduce into the trials that are widely available. How are we going to go about finding some of those?
Well, for the second year, we definitely, I mean, along with these cultivars that we have been talking about, like the old cultivars, the Watkins, and also the kind of old Durum ones, that we have also been exploring some of the modern cultivars. So we
Oh,
definitely
right.
have, yeah, of course, we have some modern cultivars because we need, it's what we always have to compare. We always need a control.
Yeah.
And obviously, I have some of the widely commercially available varieties that are also within the. And I had to say, some of the results from last year are showing that some of our modern varieties that are, I don't know if I can make some publicity of them at the minute,
Uh,
but we have some modern cultivars that are being grown
yeah,
that they
we'd
actually
probably
have an interest in architecture.
Yeah, we'd probably better not mention them by name
Those,
yet, just in case I don't know plant breeder comes down on us heavy or something like that. But again, watch this space because we'll obviously be looking to plant those out within the root ranges next year. So really exciting project to be involved in. And one of the other things that we're going to be looking at within the project is because you were talking about. The actual sort of structure of the roots and how it branches and its fibrousness and so on and so forth. Actually assessing that is quite difficult, but there is a method of assessing it, isn't there? Shovelomics, and that's something that we're going to be looking at developing. Not many people in this country really use shovelomics an awful lot to assess rooting structure, but we're going to be looking at it within the project, trying to develop a quick and easy way of being able to dig up your roots, use shovelomics to assess just what's going on there and assess a little bit more about rooting health. But I mean, we could go on for the whole afternoon talking about the truth and roots and soil and root health. But we must move on, because let's move to your next system. And I mean, actually, I think that your next topic is very much related to the first one. So it's about shifting the focus to new strategies for cost-efficient use of fertilizer and preserving the productivity of our soils. I mean, I think this largely comes back to what we were talking about earlier with biological nitrification inhibition, isn't it, Maria?
Yeah, it is.
Yeah.
Well, I think it's important. I mean, so far we have been discussing how what we are going to do and why we do it. But the reality is that we also need to change the way we look at the agricultural systems. It is what we need to focus on. We need to focus. Perhaps there is an immediate reasoning of we want to decrease the input of fertilizer because they are very expensive, because
Mm.
they get lost. It's not efficient. But the reality is what we have to focus. Is the way we look at soil. I think the main aim of what I was saying is that we need to look at soil as something that is it really sustain more than 90% of the food that we produce and eat, and it's getting lost. Soil is getting lost. So if we want to keep growing our crops, the way we grow them, we have to make a focus more on how the crop can also take care of the soil,
Mm.
and then the soil can take care of the crop. So that's perhaps the main message of it.
Yeah. And I mean, I think that, and then again, this comes back to what we were saying earlier about the we know so much about what happens above the ground and so little about what happens below the ground. Because there is, what is that thing that apparently in a teaspoonful of soil there's more life than there are humans on the planet, or something like that. Isn't that right? Um,
It's unbelievable the richness of the ecosystem,
yeah.
the life that is in the soil. Soil sustains all life and is also full of life. I mean, it's just
Hmm.
we are only touching now. There has been a lot of advance in our capability of understanding, particularly at the microscopic level,
Yeah.
what life is there. One of the earliest studies I did when I started at John Innes, we found 15,000 different species of microbes,
The 15,000 different species of microbes in the soil.
and that's
Um,
just the ones we can identify. But we know there
right.
are more.
Well, so hang on. Are you saying that there's lots of species in the soil that we still haven't yet even identified? That we don't know what they are, let alone what they do?
As this being my job, I can tell you that pretty much every week there are a few new ones. I can keep up.
Hang on, hang on. Every week you're discovering new
Oh yeah,
ones.
there are always new ones in the bibliography. You just open your Google and poof, somebody has found a few more.
I mean, that's the thing. I can see now why you're so fascinated in your job. That, I mean, it must be like going a child in a sweetie shop, and every time you go in there, there's about 10 more jars to choose from. It sounds fantastic. And what's more, I suppose what's exciting here is not only finding these microbes, but working out what they do and their various roles within this. And the more we can understand that, the more we can deliver those, important questions like how can we get more out of our crops? How can we improve that exchange within the soil? I guess is that right, Maria?
That's right. It loops back to what we were talking at the beginning. We have our crops that we need to grow for eating, and they are not in any way bad for the soil. It's just that some of them really have the capability of controlling what the
Mm.
microbes do, which, was, like you say, is not who they are, is what they do.
Yeah,
And it turns out that
absolutely.
our crops have the capacity to control it in a beneficial way. And when I say beneficial, it's beneficial for producers, for humans, and also for the environment.
I think the potential there is huge, and we're just, at the very tip of the iceberg of understanding that. What hugely important work you're doing in this area, Maria, and certainly as a farmer myself, I'm really excited to see what comes out of this and what we can explore within our own fields. That's the exciting thing. But I'm going to move on to the next one because time is rattling on. And this is about the capability to evaluate the link between crops, land management, and soil health. What are you really looking at here, Maria?
I think what is important, so it's important that we put this work and this effort in identifying these cultivars that they can do good things for the soil and then benefit from a soil that is productive. Second one, like we have said, it's kind of change a bit our view. Like, let's the crops do what they can do good for the soil and benefit from it. And the third one is like at the end of the day, our farmers and our producers are the ones that are in the field and it's their soil at the end of the day.
Mm.
So we had to give the capability to them to do the assessment on their own, to develop, we have to develop these techniques like the PES or some of the techniques that we currently use in the lab, kind of take them and into the field and give them to the people that are actually out there growing the crops so they can have the power of analyzing their soils or assessing the quality and the productivity of their soils.
Of course. Yeah, I mean the I guess that's the thing. So, a big topic of conversation at the moment, isn't it, is SFI, and the new elms that they're bringing in, the new measures that we can bring into our farms to improve the environment. I mean, it's a big topic of discussion around, are you taking land out of production that could be producing food? I mean, perhaps if we understood a little bit more about our plant systems and how our crops grow, we could deliver both, couldn't we? Both environmental improvement, soil improvement, and so on, as well as good, nutritious food. Is that really what this is all about? Do you think, Maria?
I think so, yes.
Yeah, and it's really just down to the scientific understanding of these systems. Is that right? Yeah. I mean, I just think that we often talk ourselves down or talk down the possibilities in this area, and we seem to focus far too much on whether there's food security and so on and so forth, and we look in binary terms of whether land is being productive for the environment or for crop health, but from what you're saying, it sounds as though we can do both. We can deliver good food, and we can also deliver a better environment through just understanding the science a little bit more. Is that kind of what we're talking about here, Maria?
That's exactly the message. If we can understand how the things work, then we are in a good position to act on it and make it work to everybody's win.
Yeah, absolutely. Oh, well, no, that's fascinating. And let's hope that there's going to be I don't know, an SFI payment sometime in the future for I don't know, delivering soil health through the genetics of your crop or something like that. I don't know. Let's see whether
Precisely
we
that.
can. Let's see whether we can twist a few arms in DEFRA to make that happen. But listen, thank you so much for this. It's been such a delight talking with you, Maria, because this is such an exciting area of science. And, I'm so thrilled that we've got this Truth Project and we can spend the next 3 years looking into it a lot more. But before we go, I just want to ask you to sort of look into the future, if you like. And I don't know whether there's one particular thing that you would like to achieve scientifically or whatever, or would like not necessarily you achieve, perhaps us as a society achieve, or whatever. Where is this going? Is there one particular thing that you would like to see happen that you think is possible if we have the right attitude and the right ambition to achieve it?
Well, my hope has always been. I mean, this is a big step for me, particularly having this project with you and the other partners. Is finally we have put our hands together with the same objective, and I think this should be the beginning of something that should be just the normal thing to
Mm-hmm.
do. So I looked into a future where the things I do have it's been my dream since I started working in science. The things I do, they can translate immediately into action in the field. So it's not just I publish one of my papers and it gets somewhere lost in the internet. No, it's actually I do something and then it translates into a change that move us into using the natural resources in a way that we don't destroy them. We just keep making them work for future generations. So that's pretty much how I see the
No,
future.
yeah, well, I couldn't agree more, Maria, and that's what BOFIN is all about. It's farmers working with scientists to make a real positive change. And so, all of our projects are about that. And I'm so thrilled that you're on board with Truth. And as I say, looking forward to the discoveries that we're going to be making over the next 3 years. And join us, do please, on that journey. As I say, if you go to truthproject.co.uk and have a look on there and find out how you can become a root ranger, how you can become involved. One thing I should say is that it's not just the root rangers who can get involved in this project. They're the ones who will be at the center of the project. They will be paid for the trials work that they do, and for developing these new practices that we're hoping to bring in through the course of this project. That you can also get involved as well. There'll be opportunities to send in samples and understand more about your soil. We're also going to be looking at the arbuscular mycorrhizal fungal aspects of our soil. Following on from last year's project, we had some really interesting results from that. Again, if you go to our website, bofin.org.uk, you can find some more information about that and the AMF project, or soil fungal communities project, sorry, if you have a look in that section on the website. But look, this is all we've got time for. Maria, thank you so much. It's been such a fascinating journey through your work, and through looking at the possibilities that we can open up with this. Thank you so much for joining us today.
Thank you, Tom. My pleasure.
Great. Anyway, listen, join us again on the BOFIN Buzz when I'm not sure what the next BOFIN Buzz is going to be talking about, but do join us again. Keep tuned for more of this exciting science and how we can do it to bring it into our on-farm trials. Thank you very much, and goodbye.